Good afternoon. First of all, I would like to thank the organization committee to give me this opportunity to share some of our work here. And last time I was here about 2 years ago, so it's always a great pleasure to come to Hong Kong, a beautiful city with good food. And today the weather is also very nice outside. I would like to share some data about one of our favorite subjects, which I presented 2 years ago in Hong Kong too. So today is very much a quick review and some update, and also to see what kind of obstacles are in front of us. And we started this work starting about 20 years ago, actually. Time flies. And we started this work in 1996, and I remember in 1997 is the time that Hong Kong returned to China. So exactly the nuclear transfer work, the time is a coincidence with the length of the return of Hong Kong to China. I think there's a little bit of technical difficulty with the video. Oh, here we go. Okay, great. Oh, I look at it here. Sorry. Yeah. So first of all, let's take a look at a very short video clip just to kind of understand what the nuclear transfer are about. And the nuclear transfer, the concept is very simple. We all know cells form with nucleus and the cytoplasm. So basically, the first step is involved to remove DNA nucleus from a donor cell, and then we can transfer into a recipient cell. In the clinical terms, could be a patient. There's many different kinds of techniques to actually remove the nucleus, and you can use a sharp needle or can use a blunt needle. And this step we can see is the transfer of the nucleus into the perivitally space. The key components, which most of us may not realize as a difficulty, is to integrate the nucleus into the host cell. There are several techniques can be used. We so far use electrofusion. You can also use see the virus, and also using injection method called using the piezo machine. Now, just for a frequent question, we will wonder why just to do the straight injection like ICSI. The difference is that the size. The sperm, the diameter is about 5 to 7 micrometers, and the diameter of the typical nucleus in the human case is usually larger than 20 micrometers. So using a needle at that site to inject into the egg will be lysed, degenerate right away. So for the micromanipulation technique, one of the difficult part actually is not micromanipulation, but integrate the transfer the nucleus into the host sites. That's the difficult part. Okay, so this is just a technique. Now let's review the history of nuclear transfer. The nuclear transfer really started in 1952 in frog. And we know the reason using frog is because the gametes is very big. So in 1950s, we did not have the very sophisticated micromanipulation system. So any study to involve the interplay of the nucleus and the cytoplasma, we have to do with some kind of large cells. So frog eggs is one of the good eggs to work with. I'm just wondering, why do we use the chicken eggs? Chicken eggs are pretty big. We can see by naked eye. The egg yolk, the nucleus is very big. You can even use a spoon to do nuclear transfer without using micromanipulation. Maybe we should start using chicken eggs. So these are the two pioneers studying the frog cell and working on the gametes. Very soon, the pioneer come from China, actually. The Professor Tong first studied with the fish eggs in 1963, and that was the very first success story in the fish with the cloning. And unfortunately, in the world, nobody really know much about this study because 1963, I don't know how many people here are familiar with the contemporary Chinese history. There was a period of great cultural revolution. So none of the research or study allowed to be published internationally. You're actually not allowed to use your own name. So if the work was done in the one lab, the order will be the whole lab, not each individual order. So obviously there's no first order or last order. It's the whole lab. So there was a time this work was done. In a very similar manner, in 1983 and 1986, a successful clone in mice was performed actually in Russia. At that time, it's called Soviet Union. It was successfully cloned using embryonic cells, and again, it was not published. It was not published in the international journals. But by that time, the successful cloning was done in serious species, including the first primate, the mammalian species will be in mice. Now, up to that, in the 1986, to my personal knowledge, I know that embryonic cell or plasmid cloning in sheep was also successfully done in 1986. But for some reason, I cannot find the literature, but I know personally in Cambridge by Dr. Sting Wilson and Dr. Twinkle Garland. The reason I know that is because Twinkle Garland is my former mentor. So I know they did the first sheep cloning, but it was, for some reason, was not in any literature. The real breakthrough and cloning become very well known in general public happened in 1996. That was the very first time the cloning was successfully done in dolly with a somatic cell. And so I think the real value, the revolution point in the mammalian species cloning started at the time when you actually can use somatic cells, not to use the embryonic cells or gametes. As this morning the previous speaker said, somatic cells belong to us. Germ cells belong to the God. So embryonic cells, not embryonic stem cell, embryonic cells, the cleaving breast mirrors maybe belong between the God and ourselves. So this is truly is a revolution and a big breakthrough in mankind. And at the very same time, in 1996, we started this work to do the nuclear transfer in the human eggs. We first published in 1997. The work actually truly started in 1996 when the dolly was born. But these two projects is purely coincident. We did not start the work because of Dolly, but we were thinking to do this work. That is the first time to prove that a nucleus from one cell can behave normally in a host cell, and that laid down the foundation for all the works we've been performed since 1997. In 2016, it was the first time the proof of the concept that not only the nucleus can behave normally, appearing normally in a host cell, but it the first time proved that a healthy live birth can be derived or produced through this technique in the human. And this is a first time which using the spindle transfer, which I'm going to describe the technique in a minute. And shortly, our partner in Ukraine had up to today 6 more live births using slightly different technique in a group of patients with repeated embryo development arrest and recurrent implantation failure. This is one of the first using this technique. The technique using here is a prenuclear transfer, which I will describe a different technique in the charts, which is much easier to explain. So here I would like to just describe the different kind of nuclear transfer procedure which we can use in human. Now, obviously, in the previous review slides in animals, it's almost exclusively for cloning. In sharp contrast, for the human, all the study is for how to use this technique for the clinical applications, which involve sexual reproduction, nothing to do with the cloning. So the techniques, well, kind of similar, but the material we're using is completely different. Here is 3 different kinds of nuclei we can use for the nuclear transfer. First is geminovesicle, which we studied in 1996 and 1997. Second technique we can use is a nuclear transfer on the spindle, typically we call it, which is the metaphase 2 oocytes. We also can use the prenuclear nuclear transfer, which we studied in 2003, actually not so far from Hong Kong. Our pioneer work and clinical work was actually performed in Shenzhen, which is about an hour away from Hong Kong. And I remember that was during the time when the SARS was very, very severe in China. And at that time we went to Shenzhen doing this prenuclear transfer. And I know I have only 30 minutes, but I can't help myself to tell you the story. And in 2003, you may remember SARS was in a wild epidemic in mainland China. And I myself, with my embryologist, and Showman probably know I was a fellow in New York University. We came in the whole airplane, only have 5 passengers. So there are more passengers than flight attendants because of the SARS. And the airline I took was Cathay Pacific. And this morning I got here by Cathay Pacific. So I was just asking myself, wow, 2003 I came here, it's only 5 passengers. That's because of a SARS. And the reason we came here is doing this prenuclear transfer. So you remember the 6 live births in Ukraine with our counterparts are all from this prenuclear transfer. And the first live birth we obtained in 2016 is from spindle transfer. So now we needed to see if we completed the live birth baby from geminal-vesicle transfer. This is just to show you a clinical case, which I'm going to show in a minute. I just want to quick touch the base about sequential nuclear transfer. Sequential nuclear transfer is give us the opportunity for more clinical applications. And by the way, here, I just want to skip these two slides very quickly, but just want to appreciate the difference between nuclear transfer in human and a nuclear transfer in the animal. The major difference is that in the nuclear transfer in the human, as I said, still involve the sexual reproduction and hardly have any reprogramming of the nuclear. While the cloning in the animal, it involves extensive reprogramming of the nuclear because the nuclear transfer is going to play different role as they were before, and it involves series of biochemical change in the DNA structure, which called different gene expression, which many speakers are going to discuss in more details. I just touched the base here. And we just need to look at these two lines. As you can see, the gene reprogramming in the cloning, when you inject the somatic cell, versus nuclear transfer or normal fertilization, the gene expression is very, very different. This is a normal gene expressions. So the clinical application of nuclear transfer in the human is several. One of the clinical application, as we already know, to treat a patient with mitochondrial DNA disease. And we all know that mitochondrial DNA is a small DNA molecules, and it mainly, not 100% exclusive, mainly in control of energy production. But the more study found, they probably do more than just what we know. For now, we also know that mitochondrial DNA is maternally inherited, but that's just what we know from the textbook. However, the recent study from our colleagues, I think it's already published, that actually mitochondrial DNA can be inherited by sperm. So that's something new. We all know that due to the fact that mitochondrial DNA is in charge of energy production, that's why mutation can cause multi-organ disorders. And the incidence actually is not as low as we thought. It's actually pretty high, the incidence. So the idea of the application using mitochondrial DNA and using nuclear transfer to treat the mitochondrial DNA disease is basically to take the DNA from the mutated egg into the healthy eggs. Why do we use the PGD? I will explain to you why PGD may not be an ideal option. During the last 20 years of work, and we already witnessed that, including our work and from the various groups, we can see extensive works have been done in mice, in monkeys, and in humans. First of all, it proved that spindle nuclear transfer can produce healthy live birth baby, and this just show the birth weight in the spindle nuclear transfer offspring in the monkeys, and they all develop nicely, and the heteroplasmy of mitochondrial DNA is very stable, as we can see from this line. Also, more basic study in the human eggs also proved that by doing the spindle DNA from the mature egg can actually significantly reduce the mitochondrial DNA from the donor cells. And this is a basic foundation for using this technique to treat a patient with a mitochondrial DNA mutation. For example, in this patient, we can see that this is our patient, which was the first case we did. And you can see this had 4 miscarriages, this lady from the family, from the mother's side. And had 2 boys and girls, and 1 boy and 1 girl, all deceased at 8 months and 8 years old. And this is a healthy live birth after this nuclear transfer technique. The reason we said the pre-implantation genetic diagnosis may not be an ideal option is most of patients with mitochondrial DNA disease do not produce good quality eggs and usually end up with very minimal of good quality. As you can see, this, you can't believe from a 26 years old lady what is the egg you will see, and we see quite a few patients have very, very, very poor quality eggs. So pre-implantation genetic diagnosis may not be feasible. Here you need to have enough number of eggs. And we also found that if the patient had a recurrent miscarriage or disease, the children due to this disease usually have very high level of mutated gene in their eggs. So a passive pre-implantation genetic diagnosis is just clinically not so feasible. So that's why the nuclear transfer provided new options. So this just shows how we perform the nuclear transfer. And first we're going to remove the nuclear from the patient egg. Then we're going to transfer into host donor egg in a similar fashion. Of course, the donor the DNA already removed. Then we start doing electrofusion to integrate the DNA into the oocytes. Actually, with our technique, the nuclear transfer technique actually is very efficient, and the cell construction almost reached to 100%, and the fertilization rate also optimal, and the breast formation rate also pretty good. And we are very fortunate to have male embryos, and obviously we want to transfer the male embryo. Just want to take a look at the outcome. And we can see using next generation sequencing, you can see this is between the oocytes and the embryos, and this different tissue, the mutated gene DNA is reduced dramatically. So this clearly shows that this technique is feasible. And this is a baby in the life chart. And so far, the baby is normal, and he was born April 2016. And so far, we follow up the baby, and all the criteria within normal limits. The future application of cell reconstruction in human using nuclear transfer is tremendous. For the spindle nuclear transfer and the pronuclear transfer, clearly can improve the embryo quality in the patient with mutated mitochondrial DNAs, and also for the other clinical applications for the fertility and for the poor embryo development, for the advanced maternal age. I think this will be a very important technique to add into the list of human reproductive technology. Thank you for your attention.